welcome to a new chapter. Actually redoing chapter 13 because I made some big changes, some edits to improve things. Is kind of the way that I wanted to do it. But this deals with viruses, viroids, and prions. There's some pretty substantial changes to viruses and prions from what you might see on my YouTube channel if you've watched it on there. So doing some new things. I have the photo I actually chose, the so-called father of virology, Martinez Biernik, I believe is how you pronounce his last name. So let's get started. So we're going to really get into some things. I would like to do a little bit more on some of these, but I've really changed, like made sure really getting into the types of influenza and things like that. So we're going to talk about some things in this. And we're going to talk about viruses. Now, when I was a student and I first learned about viruses, it was kind of a big deal. Okay, you had those characteristics of life that I would teach in a general biology class, that I learned in general biology, and I learned through education, is that there are characteristics of life that things that kind of are telltale signs that something is alive, and viruses on their own don't have that. And this is why we're calling them obligatory intracellular parasites. What does that mean? Obligatory means I'm required to. Intracellular, in a cell, parasite, I live off of you. They are required to have a living cell to live in to do the things they need to do. They don't have any of the things that your cells have that let them do this stuff on their own. They don't have the ability to produce ATP. They cannot produce proteins on their own. They do not have a lot of metabolic enzymes. Now, there will be some enzymes inside of them we're going to see, but they're not going to be able to do cellular respiration on their own. They're not going to be able to do things that a normal living, like a bacterial cell, will do. Now, there is debate that these did evolve from living things, but they have to infect living things, okay? Is definitely, you must have a living host of some kind, a bacteria, some kind of prokaryotic or eukaryotic cell with the telltale signs of life in it. Now, they can have DNA, they can have RNA, but they never have both in them, okay? And that's kind of big. So it could be a single-stranded RNA or a double-stranded DNA, but you never have both. They have a protein coat we call a capsid. This is kind of protective. They may have an envelope. They may not have an envelope. It's actually a lot of times a good thing if they have envelope. Like envelope viruses like COVID was, meant that I could wash my hands and give some kind of level of protection. Just one more level of protection with that virus. This is why washing your foods does not wipe out every possible infection. Like right now, it's very hard. Like if you had cryptosporosis, the on that protozoan, that parasitic thing on your food, it might be pretty resistant to washing. But if you're using soap and water, enveloped viruses are pretty easy to kind of deal with that. That is an improvement. So they have lipids. So basically, I kind of like to think of this as just this oily, kind of like a phospholipid bilayer kind of deal. Okay, there's proteins and there's carbs, just like we have in our cell membranes. There may be spikes, and this was kind of a big deal with COVID. These are used for attachment and identification of viruses. The spike protein on COVID-19's virus, SARS-CoV-2, that was what the mRNA vaccines were coding for. That mRNA sequence coded for the spike. And then our cells take that, our ribosomes read it, make the spike, our antibody sees the spike protein, then attacks the virus. Okay, it primes us to do that. All right. So let's get into this. Now, one of the things is, so I always kind of say, hey, you aren't going to get parvo from your dog or, things like that. Distemper, okay? There are viral infections you are not going to be able to get because they do not infect humans. There's a species specificity. Most viruses can only affect not only cells of maybe one kind of host, but sometimes only certain cells, like maybe kidney cells or maybe a type 2 pneumocyte or maybe a cell that just happens to have an ACE receptor on it. Okay, maybe only a CD4 T cell. Only very specific because of what proteins they have. So what can happen is you may not be able to get a particular virus like from your dog. Now, there are some viruses out there like rabies that can affect many mammals. There are some viruses that do affect only warm-blooded organisms. There was a virus I worked with called the Ambystoma tigrinum virus that affect cold-blooded organisms. If we could get it, it would be like that we have gotten Ebola, anthrax, and elephantiasis at the same time. It would be really nasty, but luckily it only affects cold-blooded things like amphibians. What I used to do was some viral testing for the state for various pathogens in wildlife with some molecular work I was doing. Host range. So what is that? Okay, so it is the type of host cells I can affect. Now, some viruses have a bigger range. They might be able to affect, let's say, mammals, or at least maybe a certain large group of mammals, because of the type of structures the cells have, the genetics of the cells, a variety of things. Why this is viruses need kind of like, say that you want to get into a room, you have to grab the doorknob and turn it. Well, for a virus to get into your cell, it has to have this protein attachment site, a binding receptor for it, so it can interact with it and get in. And there's other cellular factors as well that determine, can that virus affect you? So this is why that some viruses only affect very specific kind of cell, like liver cells, and sometimes they affect a slew of cells, and sometimes they can affect multiple species. They might affect one family in the kingdom, family, class, order, family, genus, species. Like, for example, tobacco mosaic virus can affect the family Solanaceae. So there is this range where some viruses, very, very, very narrow, some quite a bit broader. And this range usually can prevent that spread from one species to another, but it does spread. Like, could it go from bird to pig to human, or pig to bird to human, or what have you? Like how the Spanish flu did, okay? So, and then what happens is, some viruses might actually transfer genetic material to other cells. And that has happened. We're going to talk about that. And it's kind of frightening and really kind of cool. There's some frightening bits to that. And there's some really mind-blowing bits to that as well. All right, so let's just give you an idea how really small a virus is. So we know that a typical microbe, they're right around 2 micrometers. It's kind of average, okay, about 2 micrometers, let's just say, for a lot of bacteria. Now, if you look at the electromagnetic spectrum, we might say 400 nanometers to 800 nanometers, something like that, okay? 700 nanometers, 750, you'll see. Okay, I'm trying to remember the upper limit. So somewhere around that, okay? So that's your wavelength of the visible spectrum that humans can see. Viruses are much smaller than that. A virus is smaller than the wavelength of light. Now that is crazy. They're smaller than the wavelength of light, which means you can't see them with light. In order to see them, we're going to have to use something smaller than them. We do have that, called electrons. And we can take electrons and magnetic fields and use the magnetic fields like a lens and bend a beam of electrons, electron microscopy, to see these viruses. But they're tiny. And they can range, like Ebola is quite a large virus, but there's some very, very small virus. Okay? All right, so let's talk about a virion. And this is just your virus particle. In viral particles, viruses are very infectious. They infect some cell. They're always going to have some kind of nucleic acid in the middle, DNA or RNA. It can never be both. They're going to have a capsid on the outside, which is protein. This proteinaceous can protect them, but it also lets them kind of find a way to attach to some kind of thing on your cell, and they stick to it so they can dock and release. Now, they may have envelopes, and there are enveloped viruses out there, things like COVID. It's just this lipidy bit, this fatty bit, that is going to have proteins and other things on it that also help it bind to the host. There may be spikes. We call them viral spikes. Peplomere, sometimes we call them. They're a glycoprotein, and they are on there. They're kind of like the keys that we use to get into your host cell. They bind to receptors on the host cell and can let it get in to infect. And we can use these spikes as targets for antibodies to attack the virus. Like with SARS-CoV-2, we made an mRNA sequence by basically reverse engineering the spike protein mRNA, make the mRNA, shoot it into your body. Your ribosomes of your cells make the spike protein. Your immune system sees it and goes, fight this. And we can look at their coats and differences like structure, shape, morphology. Okay, and I will talk about that. For example, an envelope virus has a lipidy bit like influenza. This is why washing your hands, especially during flu and cold season, COVID season, is so important, because influenza is what we call an envelope virus, and hand washing is a good preventative. Does it stop all transmission? No. It is like if you were to take a risky behavior, okay, let's say like driving. Driving can be very deadly. You can die. Now, you wear your seatbelt. That is one precaution. Does wearing your seatbelt prevent you from dying in all cases? No. So you also obey traffic laws. That includes not speeding and behaving properly in traffic, following the traffic lights, following the traffic rules, using the right of way, making turns when you should, turning out of the right lanes, doing all the things, okay? Think of like all those things you do to make driving safer so that you don't die. Driving can kill you. Viruses can kill you. Taking precautions from driving can dramatically reduce that risk. Okay? So that's kind of an everyday example of what we can do with all the precautions we take. All add up. There's another big group of viruses called helical viruses. One of my favorites. This is that classic shepherd's crook image of Ebola. Ebola, one of my favorite, nasty little viruses. Hemorrhagic viruses, Marburg, things like that. Arrest in Virginia, Ebola. The Congolese current outbreak, lots of Ebola, really terrifying virus, hemorrhagic virus. So we'll talk about that. Here we have a complex virus. We call this a T-even bacteriophage. This is the virus that is the deadliest virus on planet Earth. This is the deadliest virus right here. It kills bacteria. And this is the deadliest. Really complex shape. Almost looks like the lunar lander. So, and then here we have the nasty smallpox. Orthopox virus, smallpox. This is smallpox. That lovely little internal structure there. Lovely little nasty little smallpox. Smallpox, terrifying virus as well. Okay. So viruses, we think they evolved from something alive. So we kind of give them taxonomy. We have family names, Viridae. A day is kind of how we end, like Ambystomatidae, for animals or something like that. So we have and the genus is something virus, like herpes virus. So herpes viridae, herpes virus, we might call it herpes and human herpes. HHV1, HHV2, and HHV3 are different kind of like herpes viruses. So viral species, they might have very similar bits to them, okay? They're common things they share genetically. They might have the same niche. What do we mean by niche? It's like, okay, what is the thing that I do? I infect reproductive bits, okay? The way they make their life, okay? They might have a similar way of making their life, like infecting nerve tissue, okay? All right, so herpes, we have HIV. That is a retrovirus, so it's retroviridae. Latinovirus, we call it the human immunodeficiency virus, which is the full name of HIV, and we have HIV-1 and 2. We'll talk about HIV, really terrifying virus. My generation, it was like, okay, this is something it's there, it exists, still raging. And this part of the world, we are able to deal with it. It's not the death sentence it used to be. Then in much of the world, it still is and is killing lots of people. Okay. Okay. Now let's talk about bacteriophages. Bacteriophages, they affect bacteria. I said they're the deadliest virus out there because in terms of total numbers of individuals killed, individual bacteria, they've been doing this for a long time. Now, when they do, so we have this thing called bacteriophage therapy. And bacteria, they kind of have three really big things they might have on. They have a cell wall, okay? They might have fimbriae, they might have a flagella. And these are all places the bacteriophage may actually attach to incorporate its genetics. So phage therapy, we use this as a treatment. And now, we have what's called lytic versus lysogenic virus. Now, I want to generate a drawing of this. I haven't gotten to yet. I've been generating some new drawings from Micro, as this is my first year. I've just completed my first year teaching the course. And I've been doing, it's usually after a couple semesters, you learn some things. Then we try something new like this lab kit. I don't like the lab kits as much. I'm definitely trying to make sure the lab kits that you guys are using aren't really hurting your grades. Because they are causing some stress. As I have to figure out the little weird subtleties about how to really guide students. But anyway, back to it. Sorry. Now, there are two what we call life cycles, lytic and lysogenic. Now, we can kind of talk about this, that other viruses do this too. You may have wondered, why is it I get a cold, I fight the cold off, and then I don't get a cold again till next season? But you can kiss somebody with cold sores. You get cold sores. which are caused by a virus. And then you get cold sores every now and again. They flare up. And you have a flare up of your cold sores. Why is that? Well, there's this virus laying dormant in branches of the trigeminal nerve. A lot of your trigeminal nerve branches are right around the mouth. And laying dormant in this nerve is the virus. Your immune system gets suppressed and it comes out around and you start getting those characteristic cold sores. Why is this? This is part of this two different kinds of life cycle. Lytic life cycle, we'll use it with phage. A lytic phage binds to the cell, infects it, its genetic material, hijacks the bacteria. The bacteria inside, you have viruses replicating inside, and eventually it pops. With lytic life cycle, it's kind of reminds me of that song. I'm trying to remember when the song came out. This place about to blow. Oh, 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 oh. Okay, it's about to blow. It's going to be lytic. Lytic, it's going to pop. It's time to shut down the lights and shut the DNA down. This place about to blow. Okay, so your DNA comes in, hijacks the cell, the bacteria grow, it pops, out comes viruses. That's lytic. Lysogenic, it comes in, the host cell's alive, the DNA that I've got gets put into the host cell's genetics, incorporated into it, put into it. We call that a prophage. The DNA of the virus is now part of the genome of the host. Now this happens to you. We have Ebola DNA in us, or Ebola has human DNA in it. Which one? There are viral stuff. Viruses have given us DNA. There are people, scientists out there. Some scientists think the reason we have skeletal muscle is because of retroviruses. They incorporated a gene, DNA, and integrated that into our genetics, called syncytium. And syncytium, our muscles called syncytial. Multiple small cells come together to make one big cell. And it is a single cell who's multinucleated skeletal muscle. So it's syncytial. There's syncytial cells in the placenta. We think we might be able to credit viruses for having placentas and skeletal muscle. And that's crazy, mind-blowing, mind-boggling stuff. This DNA gets incorporated, prophage. We then start to make that cell and release it. Host cell now has new properties. We can transduce it, and the DNA can make a new cell and all this. It gets transduced. So this cell still makes viruses, but this stays alive. And this is why you have herpes your whole life. You will have cold sores or genital herpes your whole life. And it flares up when your immune system gets suppressed. Now, what do viruses do in animals? There's kind of 6 steps they're going to do. Okay? Now, depending on your life cycle, you're either going to, the end is where you really big, kind of a few things that differs is the life cycle does some things. But viruses, what they got to do is come in, bind to the cell somehow. Okay? They got to attach to stuff on your cell membrane. So your cells have a cell membrane, a phospholipid bilayer. The cell comes in, sticks to it. And it docks, and there is some kind of virus docks to some kind of protein, kind of like a doorknob that it can use to enter. Then the virus penetrates. And that can be done through endocytosis or just fuses. Like if you're viral enveloped and you have lipid, you then open your coat. Either the virus has its own uncoating enzymes or the host cell has enzymes that uncoat. Then that nucleic acid hijacks the cell and we start producing viral DNA or RNA, viral nucleic acid, and viral proteins. It hijacks your cells to becoming a virus factory. It matures. We take these bits. It is like when you're in the kitchen, you have your ingredients, and then they come together, and now you have tacos. Maturation. We put all the bits together, and we have a virus. Then the virus is released. Now, if you want to keep the virus, we can bud it. We can just like, hey, I'm going to put this virus, put a little bud. Here we go. Release it. The cell that's infected stays, but it just releases viruses. If it ruptures and this place is about to blow, it pops and releases the virus. Okay? Now, if you're lytic, all cells, we're going to do this. So let's look at how we differ lytic versus lysogenic. Okay? Now, we're going to have the same basic steps here, except I have 6 here. I'm going to do 5 here, and I did not number these. Let me actually, why I'm at that, because this is bugging the poop out of me. I hate doing this, and I'm sorry. Never mind. I'm not going. Well, I've already started. Now I'm stuck with it. I meant to really. I like to put numbers. So there's going to be a little bit of a different thing here with these two. This is your general step for viruses, okay? So we have to attach, we have to get in, we have to make the virus bits, we got to put them together, and we got to release them, okay? Now, this is lytic. So what's going to happen is, just like, okay, you want to enter a door, you got to turn the doorknob, okay? Now, there's different kinds of doorknobs. But you turn the doorknob, or however that doorknob works. It might be one you pull down, might be one you push in, it might be one you turn, one, whatever. There's different ways to make that work. If that virus cannot bind to the doorknob, it cannot get in. So this is specific to that particular cell, to that host, to that species, to that group of organisms. And this is kind of where we really get that host specificity, okay? It can only affect very specific host cells. Now, some viruses can affect a bunch. Now, what happens is the phage attaches, then it penetrates. The genetic material gets in. It might shoot it in. Like, for example, with herpes virus. Herpes virus inside has such a high pressure. The internal pressure is greater than that of your car tires. And it just shoots the DNA into you. The DNA or RNA injects. Now it's in a bacterium. There's enzymes like a lysozyme that's going to break down the bacterial cell, let it get in. Sometimes they have something like a syringe, needle-like structure. But the virus is capsules outside, but it puts the DNA in. Now this is where it kind of like hacking a computer. Kind of like a terrorist hijacking a plane. This is my plane now. It takes over and says, this is mine, now you work for me now. It is the ultimate hostile takeover. It hijacks the cell machinery and says, you're not going to be making pancreas cell stuff. You're going to make virus cell stuff. You're not going to make bacterial proteins. You're going to make virus proteins. Instead of making bacterial nucleic acid, you're going to make viral nucleic acid. You're going to make everything the virus needs and not worry about yourself. And then we're going to put it together to make a viral particle virion here that we have. And it's put together, and then you have a bunch of these inside this bacteria. Then this place about to blow. It pops because we release lysozyme, and the cell pops, and just virus everywhere, like confetti, okay, or glitter bombs. As I like to say, glitter is the herpes of craft products. Now, these virions go out, they go out and infect other cells, and it spreads. Okay. There's a couple still like little things. So, as I said, it has to attach, okay? It has to penetrate. It has to get the genetic material in. Then it has to mature and start to assemble it. And then this place about to blow. Okay, it pops, releases the new virus. Okay. Now, this is what also some viruses do to us as well. This causes damage. The organs are damaged to the virus, lysing your cells, lysing your organ cells. Now, lytic, now there's lysogenic. Lysogenic, I'm going to add an extra thing because what we're going to do is here, we're going to have to add some bits and kind of do a little bit more extra hacking. So what we're going to do is there's attachment and penetration, okay? So just like you want to go into a room, you grab the doorknob and turn it. If it is not the kind of doorknob you can operate, you can't enter. Again, that's where the specificity. It gets in, penetrates, and ejects. Now this phage DNA gets integrated. What we're going to do is this DNA comes in and inserts into that bacterial chromosome and inserts itself in. Now becomes part of the bacterial DNA. We call this a prophage. Now that DNA, it sits there as virus, it's dormant, it's just sitting there. Now, the cell divides and all the progeny cells have that prophage in it. And many generations could go by and that's in there. And then all it takes sometimes is the right thing to happen. And I like to think about this induction. Let's say we got a sleeper agent. And the sleeper agent, they're wherever they're supposed to be. They're sitting there. And someone says, banana. And the sleeper agent says, ready to comply. And they do the thing, okay? They say the secret code word. And the sleeper agent's been activated, and the sleeper agent goes into action. This is this induction. The right event activates it. It's not dormant anymore. Now we go to lytic. DNA has been excised. It commandeers the cell. We make new cells. We go lytic. We release it. So there's this delay for a while. With lysogenic, you may release cells like through other ways. It kind of really depends, but this is how bacteria are going to do it. One of the things I also like to think about this is you've got, like it's in a zombie movie, and your group's surviving the zombies. You fight some zombies, you're like, everybody okay? Everybody's like, yeah. But one of your friends was bitten and they kept it a secret. And you're surviving together, and they're looking bad, and all of a sudden they die and turn into a zombie and try to eat you. Okay? It's kind of how I see the lysogenic life cycle. Okay? All right. So the lysogenic, You incorporate the DNA from that virus, gets incorporated into the bacterial DNA. Then we call that a prophage. When the right things happen, kind of like activating a sleeper agent, life goes on for this sleeper agent. But all of a sudden, code word, bam, activate, excise, make, do, ready to comply. Kind of like the Winter Soldier when they took the code book and read the code phrases. Ready to comply, soldier ready, or whatever. I can't remember how you said it. All right. Now, let's get into some viruses. A really big one, kills people, been very deadly, is influenza. There are three kinds of influenzas. A and B are the big ones you're going to be hearing about in the news. Okay? Now, influenza A, that's a bad one. Okay, that's a bad one. It does humans, birds, pigs, horses, other animals. Birds and pigs, that was kind of big. This is, the really bad, virulent, severe. Okay. Now, it's the large-scale epidemics, very deadly, devastating global pandemics like the Spanish flu. It's got a high, what we call mutation rate, this antigenic drift and shift, we call it, where things change. This is why we cannot get a flu shot that affects every flu. Now, there are proteins, and we said we classify that. Now, there is the flu viruses, okay? We have hemagglutinin, and neuraminidase. H1H18 and N1N11. Now, you probably heard of H1N1 flu. Very common. Big one. H3N2 is another flu type. Now, there's influenza B. This is the least bad, less severe. Okay. It's humans. Children tend to get this. It becomes epidemic like it does. This is like, it's out there a lot for the flu season. It can be severe, but it's morbidity, which how badly ill it makes us, how bad, how does it put you in the hospital, and mortality is less. A is much higher, like lays you low. We call these lineages. B, there's Yamagata and Victoria. This isn't pandemic, but so the type influenza A. That's about it. Now there is C. It's kind of like a common cold kind of deal. It affects humans relatively rarely. Asymptomatic, respiratory. You might think it's a common cold. It's not a serious thing. No epidemics, no pandemics. But it's A and B is the ones you really need to know. A is a bad one. B is, B can be okay. Now, I'm going to talk about vaccines. Something we need to talk about here, vaccines, and there are many kinds of vaccines that we have. Vaccines, what the job of a vaccine is to do. So I like to think of your immune system, okay, like a college student, like a student. Now, you remember when you went to school, you didn't know much at all. I don't even know what red is, you didn't know a lot. So you start off, you're learning to count, and you're learning numbers, and you're learning the alphabet. You start learning more and more, and after high school, you got, okay, I got a pretty regular thing. Then you start college, learning even more. Well, think of your immune system like a student. It's got to learn. And it learns through experience. One of the things when our immune system learns is it makes antibodies. And antibodies allow us to fight pathogens. Anything that can make me make an antibody to produce an immune response, that's a vaccine. Now, one group is called the inactivated vaccine. We use a killed version, or like I said, just killed. It's dead, so to speak. It's really like not a functioning. It cannot make you sick. It's just like, okay. This needs boosters like your Hep A, your flu vaccinations, your polio vaccinations, rabies vaccination. Polio was wiped out basically by vaccination. Rabies, you do not have to go get all those multiple shots with a big long needle in your abdomen, but you do need to get shots. You have to get it before this virus. The virus, even if it doesn't break your skin, the virus can replicate in epithelial cells, then get in the nerve, then travel up the nerve. And once it gets into the central nervous system, brain and spinal cord, it's it, you're dead. You have to get the vaccination before that happens. That retrograde axonic flow that happens takes not as long as you would think. We have what's called live attenuated. Attenuated, what does that even mean? In your vehicle, you have a volume control button. That is an attenuator. And you have your friend, they're trying to talk to you, and you're playing your favorite song. So you turn it down so you can hear your friend. That's attenuation. You weaken it. You weaken the volume. These give really big production, long-lasting, but they usually are 2-dose vaccinations. Okay? Chickenpox vaccine. They didn't have it when I was a child. I got chickenpox. Measles, mumps, rubella. You have the MMR. You got to have 2 of those. Smallpox vaccine, yellow fever vaccine, rotavirus vaccine. They all require 2 doses. 1 or 2 doses anyway. So they can be 1 or they can be 2, depending on the virus. Now, your mRNA vaccines. This is one of the big ones for COVID-19. What we do is it's not even a live or attenuated pathogen. It is an mRNA. Now, with mRNA, what we do at the end of mRNA, we put a poly-A tail. Every time a ribosome reads that, it clips part of the tail off, and the tail gets shorter, 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 and then it can't be used. The mRNA gets gobbled up and is gone. There is no reverse transcriptase in my genome and not in the vaccine. The vaccine cannot incorporate that mRNA. It cannot reverse transcribe it. Then there is no integrase in my genome or in the vaccine. So then we cannot integrate that DNA into our genetics. It is not a genetic manipulation tool. That is how it would happen, and it's not there. Okay? Now, COVID-19, that was a big one. There are many of these. This is actually over 20 years old, this technology. It was 18, 19 years old, I think, so it's over 20 years old. It was close to 20 years old or 20 years old. I'm trying to remember the exact age. I can't remember exactly when this technology was first developed. It was developed so that I could quickly make a vaccination. That's why the vaccinations were so quick to come out. Because we developed the technology to make vaccines quickly by doing this. Okay? Now, there is what's called a subunit vaccine, sometimes called a recombinant vaccine, sometimes called a polysaccharide vaccine, sometimes called a conjugate vaccine. They use very particular parts of the pathogen to actually give us an immune response. And it's a strong immune response. It isn't the whole pathogen, it's just a bit of the pathogen. And you can give this to people who have weakened immune systems because of the way it is. Now, these need booster shots. Haemophilus influenza, the HIB, the type B, hepatitis B, HPV, meningococcal diseases, pneumococcal, which is a nasty respiratory bacterial, respiratory infection, Shingrix, the shingles vaccine, the whooping cough, which is part of DTaP. Now, toxoid vaccines. These are my favorites. Diphtheria toxin. Corneobacterium diphtheriae, Clostridium tetani. These things kill us, make us really sick with their toxins. So what we do is we're like, hey, it's kind of neutering them or pulling the teeth out of the predator. So we got the predator. The predator's alive. We just pull its teeth out so it can't bite you. We don't go for the pathogen. We go for the toxin. Another way is you take the bullets out of the gun. Yeah, they still have a gun. There's just no bullets they can shoot you with. Viral vector vaccines. This is actually one COVID-19 vaccination. I forget the exact one. It takes a virus as a vector because viruses implant genetics and carry genetics to host cells. So you can take a harmless virus and it just sticks its stuff in there and it can help you with immunity. All right, let's talk about DNA viruses. So DNA viruses, when they multiply, they do a little bit differently. Okay, here we have a papillomavirus with its DNA. It attaches. We bring it in. We kind of might do some things where we start to make viral bits and make a new virus. We start to make new viruses. Okay. RNA-based viruses can be really weird. We have three kinds. There's a single-stranded RNA, a positive sense virus. There's a single-stranded RNA, a negative sense virus. And there's double-stranded RNA viruses. What? Okay, we'll talk about that. Single-stranded RNA, they're called the positive sense virus, okay, is you have a positive RNA. It gets translated into proteins. One of these is called RDRP protein. Do not worry about that. Let's just give it an example because that is a protein in this diagram, I believe, that our book had. It gets replicated by RNA polymerase to make what's called a negative sense RNA. That negative sense RNA now can use to make more RNA and make more viruses. See, the problem is the host cells usually have DNA in them. And the virus has RNA, and these are different molecules. So I've got to go through extra steps to get around that problem. The single-stranded RNA, the negative sense, it says, okay, they're complemented in the mRNA. They can't be translated. So we have what's called an RNA-dependent RNA polymerase. That's the RDRP. It's in the virion. We get the virus carries that. It turns the negative RNA into a positive RNA. Now it can be read. The positive RNA can now be used for RNA synthesis. See, what we're trying to do is get around these problems with, hey, that I've got RNA and my host doesn't. Now I can make the negative RNA that is needed for the cell because these things don't work the same way as we do. Influenza and measles are negative sense. The double strand is like, hey, I've got a positive strand of that is used to make messenger RNA, use that RDRP. And then you separate them and it replicates a new double-stranded RNA. And that's how rotaviruses work. Okay? All right, now this is a big one, and this is retroviruses. I'm going to do 3 really big steps for retroviruses, and then we're going to go into all the steps. So this is really kind of complicated, but I'm going to start with really some things I really need to talk about. And there's 2 things I want you guys to know about. And the first one is reverse transcriptase. Now, you know what transcription is. DNA transcribed mRNA. Okay? Well, if we reverse that, mRNA, RNA, to DNA, if we reversed it, We take an RNA molecule, viral RNA, and turn it into DNA. What are we trying to do? Get around the problem that my host is DNA and the virus is RNA. And these two are slightly different. So we're going to reverse transcribe it. We're going to integrate. We're going to make what's called a provirus. We're going to put my genetic material, the RNA of the virus, into DNA, then take this DNA and stick it into my host cell's chromosomes. We call that a provirus. Okay? Then what we're going to do is we can transcribe that, make DNA into RNA. This RNA gets packed in the virus, and bam, we got new retroviruses. HIV, very big retrovirus, okay? So that's about how this happens. And what I'm going to do is draw it out. Okay, let's draw it out and talk about it. All right, now we need two kinds of cells here. We're going to need two cells. Sorry, I didn't know if this was going to be I had to get brand new markers, okay? All right, now here we're going to have our host cell, okay? That's our host, okay? Crap, I got to close this down. Luckily, I have found out that I can fix some of the problems that I'm having with this software. A little bit, there we go. It always freezes up. Let's do this. Let's autofocus lock it. Okay, we have a host cell. Now what we're going to do, let's use blue over here. Let's have a virus. Okay, this is not just any virus. This is a retrovirus. Now this retrovirus, to be a retrovirus, what do we have to have? All viruses, remember, we have some kind of nucleic acid. RNA or DNA, but never both. This is RNA, okay? This is an RNA. This is a viral RNA inside here. And on the outside, we have our protein coat. Now, this virus, however the virus, now what we've got, okay? I'm going to make my DNA here, okay? This is the host DNA, okay? Now, we've got to get this to do like every other cell. It's got to dock, it's got to implant, and all this good stuff starts to happen. So what we're going to do, we're going to take this virus, and we're going to do the docking thing that we do like we've done in the other part of the lecture, just like we do. And this virus will take its mRNA molecule, and the mRNA is now in the cell. Now, what's going to have to happen here is, guys, this mRNA is different than the host DNA. And there's a couple enzymes that this virus snuck in with its DNA. Okay, let's talk about the first one. Very important that we understand how this works, okay? What we're going to do, guys, is we've got a thing that happens, an enzyme called reverse transcriptase, okay? And I'm going to make sure I write this where you can read it. Reverse transcriptase. Sorry, I thought I had more than that. Reverse transcriptase comes in. Now what we're going to do is we're now going to have what was this right here? Okay. This was an RNA molecule. This is a DNA molecule. All right. Now, what we're going to do with this DNA is we're going to come in and we're going to stick it in the host's genome. Now, how are we going to do that? The virus gave reverse transcriptase is a way to turn messenger RNA into DNA so that DNA now can be implanted. But it's got another enzyme, and this enzyme is called integrase. And integrase's job is to integrate this DNA into the host's DNA. Okay? Now, basically, what's going to happen is it's now part. And when we want to make new virus, you're just going to do good old transcription of that DNA, and we're going to make RNA. And now we can take this RNA, pack it, and release the virus. Okay? Drop my cat on the floor. There we go. Okay. All right. I hope that helped. Very important there. And that's what's going on here. Okay, we attach. We have to have some kind of glycoprotein in order to, like a doorknob on the door for the virus to get in. This is what determines what cell the virus can actually infect. It has to enter and uncoat. It has to get unwrapped to get the molecule in. We have to take, so with it comes two enzymes, reverse transcriptase and integrase, and the viral RNA, all come together like a package deal. The RNA gets reverse transcribed by an enzyme called reverse transcriptase. Then what happens, that means we turn our DNA into double-stranded DNA. We call it cDNA because this is kind of a special moniker we give because we're dealing with retroviral stuff. Now we go into integrate that in, and that's when we make a provirus. We need an enzyme called integrase that puts the DNA that we made. We had RNA, DNA. This DNA now gets stuck in the DNA of the host cell, of the host, integrated. We call that a provirus. Then basically you're going to start reading that. You're going to transcribe that gene that's in my genome now and make viral proteins. And that can make new viruses. I might want to put capsid proteins. I might want to put reverse transcriptase or integrase or envelope proteins or whatever I need. I put the recipe for making a virus into the genome of the host. The host now puts this virus together and it can release it, but doesn't always kill it. And then it spreads, and that virus is still free to make it for quite some time. Eventually it might kill that virus, might kill that host cell, but not before that one original cell has infected many. Okay, and that's how we do it. It has to bind, get broken in. We take the viral RNA, reverse transcribe it into DNA. DNA gets inserted to the genetics of the host cell. We call that a provirus. We transcribe that provirus DNA. We make RNA and proteins and stuff of the virus. We assemble it together, and then we release more virus. The virus spreads through the body, and this is how this works. Retroviruses, for example, can affect T cells, like with HIV. Now, if I am a cell that is enveloped, I can do budding and be released through my cell membrane. I can use the Golgi, the ER, the plasma membrane, the endomembrane system and hijack it. The viral spikes get inserted into the membrane and we put it out. You can leave that virus alone. The virus can leave the infected cell alone, and it just keeps budding viruses for a long time. Now, many viruses can be asymptomatic. You get them and they lay dormant for a long time, like cold sores, shingles. Cold sores, the herpes simplex, you get it. You can get it with nothing you've done wrong. All it can take is you were a little baby. And you're sitting in your stroller. And Great Aunt Bertha shows up. And Great Aunt Bertha got cold sores back in the 60s. And she says, "Why can't we be a little baby?" And she kisses you. And now you've got herpes simplex. Now you get cold sores. You didn't do anything. It's just Great Aunt Bertha kissed you. Okay? All right. Now, persistent viral infections, long period. These can be really fatal. From measles, you can get subacute sclerosing panencephalitis from that. Okay, long term, where you have very persistent viral infections. Okay. Now, viruses. Before we do viruses, I want to talk about things like oncogenes. What is an oncogene? Oncogene is a gene in your body that has a job to do. And that job in my cells is usually to control cell cycle. It says when mitosis should and shouldn't happen, slows mitosis down, stops mitosis, all this kinds of stuff. Okay? And so that gene that has a job to do to control mitosis, if there is a problem with that gene, and we now don't control mitosis properly, we now can get cancer. We call that an oncogene. We can activate it, and oncogenes, when activated, can make my normal cells become cancerous. Okay? Now, an oncovirus, for example, HPV, can make cells become cancerous because they integrate their DNA into the viral DNA, viral RNA, into the host DNA. Makes that cell now become cancerous. And we actually know about 10% of cancers are viral induced. HPV is definitely one well-known example of this. We used to think that viruses were the biggest cause. It's not now. Okay. Now, viruses are things that we want to culture to study and understand. So we have to grow them, but they grow in living cells. So what we can do, for example, let's say I was doing bacteria, and I had a bacterial lawn on a petri dish. And they're grown in a simple chemical medium. The media with bacteriophage and bacteria. The bacteria grow. Then the bacteria get attacked by the virus, and it destroys them. And that bacterial lawn, which covered everything, you have patches of dead bacteria. That's your plaques. It clears the lawn. It's like when you have a lawn and you have patches of dead grass. Those patches of dead grass on a bacterial lawn, which is bacteria cover the entire plate. Now the patches of dead grass, the patches of dead cells are the plaques. Each plaque is one virus that did that, and this gives us an idea. It's like a plaque forming unit. It's kind of like a colony forming unit in bacterial colonies, but for viruses. Now, if I want to study them, they have to be in living things. So we can use living animals to study immune response. To grow vaccines, we use embryonic egg. This is why some people can't take certain vaccinations, is because they're allergic to eggs. We can use cell cultures, HeLa cells. Okay, if you aren't familiar with HeLa cells, it's Henrietta Lacks. Henrietta Lacks was an African American lady. She was poor, living in the city, And during times where she did not have rights, she goes to Johns Hopkins because she knows that she can get free treatment. She had cervical cancer. And the doctor, they were like, well, trying to do some things. And while Henrietta Lacks was not awake, he goes and takes a bit of her cervical cancer cell. Turns out, for a long time, they've been trying to grow cells out of the body. And it's very hard. It doesn't work very easy. You have to have cancer cells. Well, Henrietta Lacks' cells work like a dream. And so this scientist is like, I can keep propagating Henrietta Lacks' cells. And he shares them with his fellow colleagues. And next thing you know, they're everywhere. When I was in college, I used Henrietta Lacks' HellaCell. In college, when I took cell biology. So, cell cultures is a really big thing. And Henrietta Lacks' family was trying to sue. Now, let me tell you, just think about this. All the people who use HellaCells in education, okay? And think, I got my education with it. The money I've made because of the job I could get. Doctors who got their degrees using that. People who got their degrees using it in classes. Scientists who developed life-saving vaccines and various other treatments. And then how much money the pharmaceutical companies made out of that. It is uncalculable how much money were made from HellaCells. And they did it all without her consent. Look into it. All right. Let's talk about prions. Very terrifying prions. This is a proteinaceous infectious particle, prion. Okay? There you go. Prion. All right. Proteinaceous infectious particle. Kind of where we get it. Now, what happens is, guys, how do we get it? Now, we usually ingest it. Okay? Like, hey, I ate brains. Surgical instruments can transport it. It can be from a transplant. One is like spongiform encephalopathies, like sheep scrappy, Creutzfeldt-Jacobson syndrome, Gertzmann-Straussler, Schenker, I believe, fatal familiar insomnia, mad cow, wasting disease, all these. Now, on cell surfaces, there are proteins. And proteins can be misfolded. Not all misfolded proteins are prions. Not all misfolded proteins are pathogenic. Some just don't work. But when it gets in. So let's say we have a normal protein that does a job on a cell called PRPC. If it becomes a version due to a structural malfunction, PRPSC, the scrappy protein, gets on the brain cells, forms plaques. This is kind of what happens in Alzheimer's with the tau proteins and all that, those amyloids. That's what things like drugs like Leqembi is. Leqembi takes these misfolded, messed up proteins in the brain of Alzheimer's patients and acts like an antibody attack system. Now, there's normal and infectious shapes. Like all animals have these proteins. Not every single protein becomes prion. Okay? PRPC. That's the normal one. It's found in the brain. PRPSC. Now they have the same amino acid sequence, but we fold them differently. A rogue PRPSC finds a normal one. And it's like a template. Then you get a disease shape. It's the disease shape. It's not just any misfolding. It is the wrong misfolding. What do I mean by that? You got to fold up in a particular way that makes you pathogenic. Now it's infection. It's infectious. It makes a chain reaction. Proteins misfold. They can clump together and build up, called amyloids. And this is what happens in Alzheimer's. That build up makes holes in the brain, so we call it spongiform. Holes like a sponge. And it's transmissible. So there is a version of Creutzfeldt-Jacobson syndrome called variant Creutzfeldt-Jacobson syndrome that can be transmitted by bites. It's like mad cow disease became mad human disease, became mad zombie disease. So that's how we actually get the infectious death from misfolded proteins. Not all misfolded proteins become prions, but they can if the right things happen in the wrong way. Or is it the wrong things happen in the right way? If the very specific things happen, they can become. And this is why a lot of these diseases you get from eating brains, like kuru, things like that. Now, I want to talk about plant viruses a little bit. Plant viruses, they just get in, like if an insect bites and chomps on a plant, virus can get in through the cuticles and all that of plants. Viroids, they're actually RNAs that can become plant pathogens. It's just a RNA that becomes pathogenic. The potato spindle disease, potato spindle tuber disease. All right, guys, that is it. I really hope you enjoyed this. I really took a lot of time to try to make a better virus explanation for viruses and prions, really explaining the differences in kind of viruses you might see. Thank you, guys. I hope you enjoyed it. I hope you enjoyed what I give. Thank you, and I will see you guys in the next one.